Welcome back to another Explainer. Today, we're diving into literally one of the greatest scientific detective stories in the history of biology, uncovering the actual structural blueprint of life. For decades, scientists knew that traits were passed down, but they had absolutely no idea what physical substance was carrying that crucial information across almost all living things. Well, except for RNA viruses, which we later figured out in 1956 use RNA instead. But for everything else, it was a total mystery. Let's find out how we cracked this universal code.
Here's our game plan for today. We'll start with the mystery of inheritance, move on to finding the genetic material, break down the anatomy of a nucleotide, and finally, explore that iconic double helix structure. Section 1. The mystery of inheritance. Picture this. It's the 1920s, and scientists are completely baffled by the physical nature of the gene. They're studying microorganisms, they're watching traits get passed down, but the actual molecular vehicle driving all of this? Total black box.
This timeline maps out the whole investigation for us. It really tracks how a series of brilliant scientific minds patiently narrowed down the prime suspect over the decades. It wasn't some sudden eureka moment, It was a grueling, step-by-step process of elimination that took almost 30 years from the very first clue to the final, undeniable proof. Our first massive clue drops in 1928. Frederick Griffith was working with Streptococcus pneumoniae bacteria, and he discovered something wild. He found that heat-killed lethal bacteria could somehow transfer a substance to harmless bacteria, transforming them into a deadly form. He'd inject mice with the harmless R strain, and they lived. He'd inject them with the dead heat-killed S strain, and they lived. But when he mixed the two together, the mice died, and he actually recovered living, lethal bacteria from them. Some mysterious transforming principle had literally jumped from the dead cells into the living ones.
Section 2. Finding the genetic material. So what on earth was this transforming principle? Let's move to the next phase and see how the detectives started ruling out false leads in the hunt for the molecule of life.
It was basically the ultimate biological process of elimination. By 1944, Avery, MacLeod, and McCarty took the debris of those dead cells and systematically destroyed one chemical component at a time. They got rid of polysaccharides, lipids, RNA, and proteins. And guess what? If they destroyed the proteins, the transformation still happened. If they destroyed the RNA, it still happened. It was only when they destroyed the DNA that the lethal transformation finally stopped. It was our very first proof that genes are made of DNA.
But a lot of scientists were still super reluctant to accept that a simple molecule like DNA was the answer. They really favored complex proteins. So in 1952, Hershey and Chase used the T2 bacteriophage, an ordinary kitchen blender, and radioactive isotopes to get the ultimate forensic evidence. They tagged the viral DNA with radioactive phosphorus and the viral protein coat with radioactive sulfur. When the virus infected the bacteria, the sulfur stayed outside, but the phosphorus went inside to dictate reproduction. Boom. The case was definitively closed. With the historical whodunit conclusively solved, we arrive at a really pivotal question. How can this seemingly simple chemical act as the comprehensive blueprint for the huge linear chromosomes in eukaryotic cells or the circular genomes in prokaryotes? Oh, and let's not forget the extra chromosomal DNA in your mitochondria and chloroplasts that follows that fascinating non-Mendelian maternal inheritance. How is this universal material actually built?
Section 3. Anatomy of a nucleotide. To truly grasp how this blueprint holds information, we have to zoom all the way in to the atomic level. We need to look at the individual building blocks.
So, the crucial takeaway here is that DNA is just a long, unbranched polymer chain made up of monomer units called nucleotides. Think of it like a chain link fence, where every single link is a three-part structure. A 5-carbon sugar, a phosphate group, and a specific nitrogenous base that basically acts like a letter in a genetic alphabet.
Look closely at the sugar part, deoxyribose. The absence of a hydroxyl group at the 2' position is a total game changer. It gives DNA vital mechanical flexibility. This tiny atomic omission lets eukaryotic linear chromosomes pack incredibly densely. It also allows bacterial circular plasmids to contort into all sorts of wild conformations without snapping. Whether that's a relaxed circle, nicked open, completely linear, or even a highly twisted supercoiled structure.
Okay, so how do we link these individual blocks together? To actually carry instructions, these floating nucleotides have to be permanently fused into an unbroken line. And this involves a really elegant, highly specific chemical reaction.
Breaking down the chemistry, we see the creation of strong, covalent phosphodiester bonds linking the 3' carbon of one sugar to the 5' carbon of the next. The nucleotides actually arrive packed with energy as triphosphates. They snap off 2 phosphates to power the reaction, release a little bit of water, and lock firmly into place. This forms a crazy resilient sugar-phosphate backbone that keeps your genetic info safe and sound.
Section 4. The double helix structure. We have a solid chain now, but intact DNA actually exists as 2 strands wound around each other. Now, depending on the pucker of the ribose sugar, DNA can adopt different shapes, like A form, which has a 3' endopucker, or the much more common B form with its 2' endopucker. Let's look at the classic B form architectural masterpiece that James Watson and Francis Crick solved in 1953.
The physical dimensions of this thing are incredibly precise. The helix has a diameter of exactly 20 angstroms, featuring 2 antiparallel strands. They're pushed to opposite sides by the repulsion of those negatively charged phosphate groups. Because the strands run in opposite directions, one 5' to 3', the other 3' to 5', they zipper together perfectly. Adjacent base pairs are separated by just 3.4 angstroms, completing a full twist just over every 10 base pairs, or 35.7 angstroms, and creating a distinct major and minor groove along the outside. It's molecular architecture at its finest.
And what holds these 2 twisting strands together right down the middle? Hydrogen bonds. They strictly follow the rule that adenine always pairs with thymine, sharing exactly 2 hydrogen bonds, and cytosine pairs with guanine, sharing 3. This strict chemical complementarity is the absolute secret to how DNA perfectly copies itself every single time a cell divides.
But, as robust and uniform as this whole structure is, it's not invincible. Occasionally, an adenine or cytosine will shift into a rare shape called a tautomer. When that happens, they mispair, completely bending the foundational rules we just talked about. For just a fraction of a second, the chemistry fluctuates, and suddenly an adenine pairs with a cytosine instead. If the DNA replicates while this temporary shift is happening, a permanent error gets locked right into the newly formed strand.
Which leaves us with this final, kind of mind-blowing thought. How do these tiny, rare structural misincorporations in the blueprint ultimately drive the massive engine of evolution over millions of years? Think about it. Every natural variation in life, from the color of your eyes to a bacteria developing antibiotic resistance on a tiny plasmid, begins with these microscopic deviations in this brilliantly structured molecule. Thanks so much for joining me on this explainer, and keep questioning the science that builds our world.